Good morning, good afternoon, good evening to everyone, and welcome to a new webinar of the Global Plant Council in collaboration with the Plant Journal. My name is Isabel Mendoza, and I want to share a little bit about the logistics for today. So attendees, you can use, as always, and people have already started using the chat box in order to communicate with either everyone or with us, the panelists. But I will ask you to use the Q&A tool for sending your questions, because questions can get a bit lost in the webinar chat. So this time you will be able to submit your questions and also upvote for questions from other people, and that will help us organize and pick up these questions for the Q&A faster. The Q&A will happen at the end of the session, so it will be a great help for us if you mark your questions with the name of the speaker you want to address this question to. So today, as a moderator, we have in the room Professor Katherine Denby. She's a professor for sustainable crop production at the University of York and also editor-in-chief for the Plant Journal. So Katherine, the floor is yours.
Great, thanks Isabel, and thanks everyone for coming. And Isabel says, yeah, welcome to this second Global Plant Council and Plant Journal webinar. And we've designed the webinars to sort of enable researchers around the world to hear about some of the exciting science and enable authors of Plant Journal papers to share their work. So the speakers are not necessarily talking about work that's in the published in the Plant Journal, but they're some of our recent authors. And I just wanted to highlight that the Plant Journal is part owned by the Society of Experimental Biology, which is an international society covering plant, animal and cell science. So part of the profits from the journal go back to support scientists through meeting funds and travel funds, etc. So just to thank you, because publishing in society-owned journals helps support science, not just the publishing houses. So for this webinar, we've chosen 3 authors who contributed to a special issue last year on plant engineering biology, and I'll put the link in the chat when I stop talking. And I think you'll agree that they cover really exciting, diverse aspects of synthetic biology. So I hope you find today interesting and stimulating. So without further ado, I'm delighted to introduce our first speaker. So Dr. Adil Khan. Adil's a postdoc at the University of Western Australia and in the ARC Centre for Plant Energy Biology. And the focus of his work is developing CRISPR-based genetic circuits to program plants and enable them to respond to different environments, sense and respond to environmental signals with applications in sustainable agriculture. And he's pioneered new approaches to developing programmable gene expression in plants, and today he'll be telling us about synthetic gene circuits for programming plants. So over to you. And just to say, as Isabel said, put your questions in and we'll take questions at the end of all the speakers. So Adil, over to you. Thank you.
Thank you, Professor Katherine, and thanks for having me here. I'll share my screen. All right. Good day, everyone. Today I'm going to talk about some of our recent work regarding the synthetic gene circuits for programming plants. So for a very long time, we have been engineering plants for improved growth and yield, resistance against abiotic stresses, for example, heat and drought, resistance against biotic stresses, for example, bacteria and viruses. And this is usually done through the introduction of a transgene into the plant genome. it works fine, but it usually comes with a cost, and that is the metabolic burden, gene silencing, and the deleterious effects on the plant's performance. So if you take a look to the literature, the current tools that are available for engineering plants are usually the strong constitutive promoters. For example, the 35S promoter, which has been widely used in plants. We have the cell type specific promoters, and we do have the inducible systems. For example, the most widely used ones in plants are the dexamethasone and the estradiol inducible systems. However, these different tools have some limitations, and they are, for example, the strong constitutive promoters, they are non-specific and are commonly silenced. The cell type specific promoters, they mostly can't respond to additional conditional inputs and are constrained by their natural range. The inducible promoters, they are mostly ubiquitous, but they can't respond to multiple variable inputs. So we need new tools for more sophisticated control of gene expression so that we are able to engineer smart plants. They are able to respond to multiple input signals. So that could be chemicals, that could be environmental cues, or that could be symbiotic factors. And then the plant should be able to produce a response when and where specifically it is required. So how it is possible. So one of the possibility for this is the use of synthetic gene circuits. Some of you might already know about the synthetic gene circuit. Synthetic gene circuits. Some of you might be wondering, what are these synthetic gene circuits? So they are similar to the electronic circuits found in the electronic devices, your computers, smartphones, and all the electric appliances. So what are these synthetic gene circuits? So they are composed of interacting biological parts such as DNA, RNA, and proteins. So if we take the example of a promoter and put a gene downstream of it, so in ideal circumstances, this gene will be switched on all the time. For example, this promoter driving the expression of a repertory gene, so this will be switched on. Now, if we have a repressor that is targeted to this promoter, right? So what will happen? This repressor will interfere with the promoter and will switch it off. And as a result, the reporter gene will be switched off. So this switching off of the constitutive promoter by a repressor is similar to an electronic NOT gate. When we have the input 1, the output is 0. Now, consider a scenario where instead of one repressor, we have two different repressors, repressor A, repressor B. And as I explained, that in ideal circumstances, the promoter will drive the expression of the repertory gene, right? So when we have two repressors, in their absence, the promoter should be able to express the repertor. However, in the presence of input A, input B, or both together, this promoter will be switched off, and as a result, the repertory gene won't be expressed. So this is similar to an electronic NOR gate, which requires two different inputs, input A, input B. And the output will only be produced when both input A and B are not present. However, in the presence of A, B, and both together, the output will not be produced. And this NOR gate, we call it as a universal gate because it can be layered to make complex circuits. And this is something I will highlight later during my talk. So to date, in plants, people have used different tools for creating these synthetic gene circuits. One of them is recombinases. Some of the work we have done in this regard, DNA binding domains have been widely used. and then the CRISPR DKS9 system. So today the focus of my talk would be the use of the CRISPR DKS9 system for creating synthetic gene circuits in plants. So to talk about the experimental design, then how can we create a NOR gate using the CRISPR system? So the whole idea was to take a promoter that is driving the expression of a reporter gene. Here is a luciferase gene. And the idea was to target it with CRISPR DKS9 using 2 different guide RNAs. Here, I would like to mention that usually the CRISPR-Cas9 system is used for gene editing. Here we are using the deactivated version of the Cas9 enzyme, which has lost its catalytic activity. So what will happen in the absence of these input guide RNAs, the promoter will drive the expression of the luciferase, and ideally we should get a signal, right? When we target this promoter with 2 different guide RNAs, input guide RNA A, B are both together. So the DKS9 will bind to the promoter, and as a result of the deactivated Cas9 with the RNA polymerase, this phenomena is called the CRISPR interference. The transcription will be stopped. When we don't have the transcription, there won't be any translation, and as a result, the gene will be switched off. Now, to implement this strategy, what did we do? We took 2 different promoters from plants. One is the widely used 35S promoter, and the other one is the TCTP promoter from Arabidopsis. And what did we do? We introduced unique guide RNA binding sequences for these 2 different guides upstream and downstream of the Tata box by eliminating the native sequences. And then this engineered promoter is then driving the expression of the luciferase. Now, because when we work with plants, so it takes a lot of time to generate stable transgenic lines, so we needed a fast platform, whether the strategy is working or not. So for this purpose, for this fast platform, what did I do? I optimized the Arabidopsis protoplast system. Protoplasts, these are the plant cells without the cell wall. So what did we do? So we isolated protoplasts from 4 to 5 weeks old Arabidopsis plants from the leaves of the Arabidopsis, and then transfected them with our constructs. So the construct design is that the engineered promoter is driving the expression of the Granella luciferase, where a constitutive promoter, for example, the NOS, is driving the expression of the firefly, which serves as a normalization control, right? So when the protoplasts are transfected using the polyethylene glycol, we do it in the 96 well plate in a high throughput manner, and then we perform the dual luciferase assay. So here I would like to mention that we are not measuring the transcript abundance. We are relying on the catalytic activity of the luciferase enzymes. This protocol is available online. Feel free to use it if anybody need some guidance in this regard, because we get our results within 24 hours once we have transfected the protoplasts. So to determine whether these engineered promoters should go back here, these engineered promoters that we have created should still be able to drive the expression of the luciferases in the absence of the DKS9, because here we have engineered them, so we wanted to know whether they should still be able to drive the expression of the downstream gene. So we transfected our constructs into the Arabidopsis protoplast, then performed the dual luciferase assay 24 hours post-transfection. And then here are the results. So we generated a wide range of different strength of promoters, and we generated 2 libraries. One is the 35S best, and the other one is the TCTP best. And all these promoters, they contain unique guide RNA binding sequences for the DKS9 binding. And here you can see the strength in the absence of the DKS9. The next step for us was whether these engineered promoters can be used now to create functional NOR gates in the plant. So what did we do? We took two different promoters from each library. For example, we took the engineered promoter number 1 from the 35S library and the TCTP engineered promoter from the um, the engineered promoter number 3 from the TCTP best library. And then we tested it in the presence of the DKS9 whether we can make functional NOR gates. So here are the results in the Arabidopsis protoplast. Whether it is the TCTP promoter or the 35S promoter, you can see that in the absence of any guide RNA, the promoter is active in both cases and can drive the expression of the reporter gene. However, in the presence of input A, B, or both together, in both cases, we can see a very significant amount of repression. So, absence of any input signal, the promoter is active. In the presence of either input A, B, or both together, you can see that the reporter gene is repressed. So we created functional NOR gates in the Arabidopsis cells. Now, those who are working with the CRISPR systems, they might know that usually the guide RNAs are expressed from the POL3 promoters, and people usually use the U6 promoters. However, our main aim was to provide more sophisticated spatiotemporal control of gene expression in planta. And for that, we thought that the guide RNAs need to be expressed from cell type specific or inducible promoters, which in most of the cases are POL2 promoters. So it requires a bit of engineering. So what did we do? Is in order to express the guide RNAs from the POL2 promoter, we took the 35S promoter to drive the expression of the guide RNA as a proof of concept. And for this, we tried 3 different approaches: the ribozyme, the tRNA, and the CSY4 system. The ribozymes, so we tried it. So the design was you basically flank the guide RNA sequence upstream and downstream of the hammerhead ribozyme and the HDV ribozyme. And because ribozymes are the self-catalyzing RNA-based enzymes, so they can cleave themselves off, and the mature guide RNA is produced. So here you can see we could create a functional NOR gate that in the absence of the guide RNA, we could still see the promoter is active. In the presence of input A, B, and both together, we could still see the reporter gene is repressed. However, the amount of repression we were getting was not good as we saw with the U6 promoters. Then we moved on to the tRNA system. The design is exactly the same, where you take the tRNA sequences and put them upstream and downstream of the guide RNA sequence, and then take the whole sequence, put it downstream of the 35S or the POL2 promoter. And then because of these native enzymes, the RNAs P and Z, the tRNA sequences would be spliced and the mature guide RNA would be produced. Here we could see like functional guide RNA, which could produce NOR gate functionality in the plant cells. And you can see we were getting like decent amount of repression. However, the tRNA sequences, they contain native binding sites for the RNA POL3 promoter. And at the same time, we were getting both RNA POL2 and POL3 transcription. So therefore, it was not a good choice for us. And then we moved on to the CSY4 system. The CSY4 is a small CRISPR-related protein which targets 20 RNA base pair target in the mRNA transcript. The design is exactly the same. We provide the CSY4 binding sequences upstream and downstream of the guide RNA sequence. But for this one, we need to produce to express the CSY4 protein as well. So the whole sequence is put under the POL2 promoter. When the CSY4 is expressed as well, the guide RNA sequence is spliced and the mature guide RNA is produced. And here you can see that regardless at the 3' sequence, there is a 20 base pair remaining sequences attached to the guide RNA, but it doesn't affect its performance. But as we don't have the repression issue as we saw with the ribozymes, or we don't have the POL2, POL3 issue. So this was a functional NOR gate, and we were quite happy with these results. And then we thought we can move on to see whether we can link it to some environmental cues. So here are once validated these results in plant protoplasts, then we wanted to move and tested the functionality of these circuits in stably transformed plants. So here we link the guide RNA expression to heat-inducible promoters. Instead of the 35S, we are now using the heat-inducible promoter. We stably transformed Arabidopsis plants, and here are the results in the cotyledon. When we don't have any input, the promoter is active, driving the expression of the luciferase. And when we have the heat input, the reporter gene is significantly repressed. Here, I would like to mention here that we are measuring now the transcript abundance, not the dual luciferase activity, because these were coming from the stably transformed plants. And we performed the qPCR. Here are the results from the roots. Similarly, in the absence of the heat signal, higher activity of the promoter. In the presence of the heat signal, a significant amount of repression in the roots. And then we tested the reversibility in mature leaves of this circuit. So here you can see that when we provide the heat treatment, post heat treatment at 6 hours, the circuit performance is at its peak. So we can see significant amount of repression. And then post 24 hours, the circuit start reverting back to the normal state. And after 48 hours, it is reverted back to the on state, and then it becomes stable 72 hours. So this was, to our understanding, the very first demonstration of the CRISPR-I-based circuits, where we showed the reversibility of the system in stably transformed plants. Now, we have validated this system in Arabidopsis, which is the model plant. So we wanted to see whether we can use these the NOR gates for layering the circuits where we can perform the complex computational logics. And for that, as I explained throughout my talk, that we used a promoter that has the unique guide RNA binding sequences, which are targeted by 2 different guide RNAs. And then this promoter is driving the expression of the reporter. So now, if we replace this luciferase with another guide RNA, and then this guide RNA is then targeted to another layer of the promoter, layer 2, then that is driving the expression of the luciferase. So ideally, what will happen that in the absence of the input A and B, this promoter will be free to drive the expression of the guide RNA-C, which is then produced to target the layer 2 promoter. And as a result of the CRISPR interference, the luciferase will be switched off, right? However, in the presence of input A, B, or both together, this promoter will be occupied. The guide RNA-C will not be produced. And then this promoter will not receive any input signal. And as a result, in each condition, the luciferase gene would be expressed. So we tested the performance of this programmable OR gate in Arabidopsis protoplast, and here you can see results. And when we don't provide any input guide RNA, the guide RNA-C is expressed leading to the repression of the layer 2 promoter. So we can see significant amount of the luciferase. However, in the presence of input A, B, or both together, the guide RNA-seq is not produced. And as a result, the layer 2 promoter is not receiving any input signal. And we can see higher activity of the luciferase here. So this was just a demonstration of the OR gate. We went on further to create programmable multi-layer gates where we arrange these NOR gates into different layering architecture. So we created N imply B, B imply A, and AND gates where we connected 3 different NOR gates, and we could see the desired computational logics in the plant cells. Now, because all this data was coming from the Arabidopsis, which is used as a model in plants, so then we moved on. We wanted to import these circuits into different plant species. So I have some data from moss where we tested the fully functional NOR gate in moss protoplast. We also tested the performance of the one input NOR gate in wheat, and we also tested in brassica. So all these studies were performed in protoplast at 24 hours post-transfection. So without any modification, we could just implement them into these plant species, which is quite promising that without any modifications, these circuits could be imported into different plant species. So in summary from this CRISPR system, so we have developed the CRISPR I-based circuits, which are modular, compact, programmable, reversible, and all sensitive to the input signal strength and functional in various plant species. This work was recently published in Nature Biotechnology. And because now we have this technology in hand, so we are using it in our recently established ARC Center of Excellence in Plants for Space, where we are using these circuits for programming plants for space. So the whole idea is to use this technology for creating multi-phyte programmable plants when and where specifically we want to produce some biomedicines or biomaterials. So that is the whole purpose of the Center of the Excellence. Now, I would like to provide a brief introduction to some other work that we have been working. The other is because all this. Yes.
Sorry, you are over time.
Just this last slide and then I'm done.
Okay, all right.
Yeah. Yeah. So we have developed a new CRISPR split DKS9 activation system. Gabriel, who is a recently graduated PhD student now as a postdoc in the lab. So this split DKS9 DKS12S system, so we could achieve up to 400-fold activation, which to our understanding is the highest to be reported in plants. This work is currently under the preparation for the publication, and then we are also involved in the development of the plant synthetic chromosome, which is funded by ARIA from the UK, and is a team effort between UWA, University of Cambridge, Macquarie University, and a partner form. So I would like to conclude my talk here. Some future developments that we can talk about some other time. And yeah, so this would not have been possible without team effort and a lot of the different funding agencies who have been involved. Thank you.
Thanks very much. That was great. Very clear. And really, yeah, interesting how you can layer them up. Just to remind everybody to please put your questions in the Q&A, and we'll do questions at the end. So yeah, thanks to Adil again, and then also just like to introduce our next speaker, who is Dr. Jenny Molloy. So Jenny's a senior research associate at the University of Cambridge. She's in the Department of Chemical Engineering and Biotechnology. and also is director of the Open Bioeconomy Lab, which I'm sure you will see why when she gives her talk. So she's pioneering sort of open source biotechnologies to try and make a global bioeconomy that's more sustainable, but also more equitable. So she focuses on developing open source tools that can expand access to these synthetic biology capability in low resource contexts, and has led major international efforts to make all these tools more affordable and widely used. So today Jenny will be telling us about that in a talk entitled Taking Synthetic Biology Global, Open Source Tools for Equitable Access to Advanced Biotechnologies. So thanks for joining us, Jenny, and I'll hand over to you.
Great, just allow me to share for 1 second. Hopefully this will start immediately. Great, thank you for the introduction. So I'll be talking today, as mentioned, a little bit about the accessibility of some of the technologies we've just heard about, so synthetic biology tools, also CRISPR technologies. So just to preface the work that I do, this is the sort of pretty recent state of publications around synthetic biology. And there's a very clear disparity in where those publications are coming from. So we see far fewer publications from low and middle income countries, excluding China, which is classified as a middle income country, but is a very prolific synthetic biology publisher. So there's many reasons why we might see this disparity. Science funding in general is very unequal across the world. But as we probably all know as biologists, biology can also be an expensive discipline, especially molecular biology and synthetic biology requires labs, equipment, reagents, consumables. And so this kind of exacerbates the existing inequities in access to the necessary technologies to do the type of work that Adil just presented. And so I've been working with colleagues in my research group since 2018. This is Lenshina Agbor, who was one of our first researchers in Cameroon. We set up a lab there, and she went on then to do a PhD in the UK. And this is just her reflecting on her experience from moving from the context of the Cameroonian lab to the UK, and just the kind of gap she felt in terms of her research output and what she could accomplish in the same space of time was incredibly wide. And so a lot of our work is trying to support researchers like Lenshina to try and lessen that gap and see where we can support advanced biotechnology taking place really all over the world. And so this is an issue that's received quite a lot of coverage, even just recently. So putting aside COVID, where we all saw the kind of challenges around distribution of diagnostic reagents and other biotechnologies like vaccines and therapeutics, the scientific supply chain is being highlighted by funders and foundations like the Science for Africa Foundation, published a commentary 2 years ago, and also held a meeting last year all about how the lab supplies market is kind of what they describe as crippling African science. Also, just before Christmas, the Inter-American Development Bank published a report and a commentary in Nature Biotechnology about challenges in Latin America around the import of biological research materials and how much that is a challenge to biotechnology in the region. And an effort that I'll talk about a bit later, Reclone, is a kind of an attempt to address this. And we had a piece in Science when we started a couple of years ago to look at how we, in a Latin American context, in this case, can support access to essential lab materials. And that work is supported by the Chan Zuckerberg Initiative. And so basically, most of my work addresses this idea of the supply chain for reagents. And the reason for that supply chain being problematic is this map. So if you look at the dark blue areas, this is where laboratory agents are exported from. That typically means that's where they're manufactured. It's not an ideal proxy for manufacturing capability, but if we look, we can see that there's a lot of export from Northern Europe, North America, and parts of Asia, but very little export and indeed very little manufacturing of these reagents across most of Africa, most of Latin America, and also parts of Asia as well. And so the challenge, particularly for laboratory reagents and particularly molecular biology, is that many of our reagents like to be cold. and therefore they're being manufactured in one place and then having to be shipped via a cold chain across the world, which is often very expensive, it's unstable, and the other challenge is it's very environmentally unfriendly. We're shipping like solidified carbon dioxide across the world on a daily basis to sustain the scientific enterprise, and this is challenging. And so I'm sure many who are on the webinar today have experienced challenges, but if you speak to anybody who works in these regions, they'll have stories of how this has failed them, and reagents have arrived at great expense only to be inactivated and unable to be used once they reach the lab. So this is one of the challenges. Obviously, there are many other challenges, as I already mentioned, around why there are disparities and inequities in scientific access and scientific output around the world. But this is the one that, as a molecular biology lab, we've chosen to address. And so one thing is, I already mentioned the kind of unreliability and the cost. But actually, I think one of the biggest factors is really the time to delivery of those reagents. So I'm fortunate when I work between Italy and the UK. In both places, our kind of standard tools of the trade, DNA polymerases, molecular biology reagents will reach the lab bench in a few days. For our colleagues in Cameroon, 4 weeks was a good day, but it was often much longer. And so if you think about the time that it takes to do science, and I'm not, although I have worked with plant scientists for over a decade now in sort of the synthetic biology field, I'm not a plant scientist. I now work entirely with microbes, and one of the reasons for that is that it's quite fast work. Those of you who are working with plants will know that it's not. I'm a zoologist by training and used to work with mosquitoes, and so biology is slow. And if every time you need to order new primers, change around your protocols, you're waiting 4, 8, 12 weeks for those materials to arrive, you can imagine the impact that has. So I say this just because I think, in a sense, my work and the work of the communities that I have established, we're really addressing a very mundane issue. But what I believe is that it actually has very profound implications for who can do science, the type of science that they choose to do. And that is a kind of the bigger picture. And also for the scientists themselves, many frustrated scientists who are kind of like questioning their choice of career in some countries because they are so hampered by some of these logistics issues, basically. And so we have this high import dependency, and this is a particular challenge in the global south. I already explained the sort of challenges there, but that really leads to this kind of lack of autonomy for researchers to meet local needs, because they're often dependent, sometimes even on international collaborators sending them things that they need. And so we have this stifling of innovation effect because of these long iteration times. And so this is basically the hypothesis and the reason that we try and develop local manufacturing of some of these reagents as one intervention, which may not be the answer in all cases, but we're exploring the context in which it could be the answer. So the idea is this, the limited R&D and manufacturing capability challenges the chance that biotechnology and the scientists, the biotechnologists, have to thrive, and also not just biotechnology, but of course basic science and discovery-based research as well also depends on these tools. And so our approach to this work is to develop accessible product and manufacturing technologies. I'll describe a couple today. But technologies are kind of useless unless somebody is actually supported to use them. And so we also have a big focus on building communities. I already mentioned Reclone, which is our major kind of community driven effort to bring scientists together around this idea. We also support enterprises. So not everybody wants to manufacture enzymes in their lab, and indeed the convenience of buying from a company is a major one. And so I'll talk later, but one of the reasons that we use open source approaches is that those are then fully available to companies to develop and sell the enzymes that they produce from our plasmids or with our protocols. And we've supported companies in Indonesia, in Brazil, in South Africa, in Chile with materials and kind of guidance. We set up a company from scratch in Cameroon. I won't talk about that due to the limits of time, which was an interesting effort. It ended up not working entirely as a business, although that's a longer story, but it was the tech transfer aspect of it was very successful. And so in that case, there really is there is the scope to have these sort of distributed manufacturers around the world. And then, as always, it's not the technology that's the really hard part, it's the people and the kind of like building a business and all the rest of it. Anybody in the room who's tried to do that before knows that there are many, you know, in contrast, science is in some ways more predictable than trying to do these things. But so the overall goal is that we can increase resilience and autonomy through a more distributed supply chain. So bringing local manufacturing to the table as a partial solution to some of these challenges. We mostly work in low middle income countries, but some of the technologies that we build upon, there's also interest in kind of really distributed supply chains for kind of in the remote wilderness. I have colleagues who have manufactured enzymes in Tierra del Fuego in Patagonia for doing kind of diagnostics work there, or it's environmental surveillance. There's also interest in kind of distributed manufacturing of biotechnologies in crisis zones and also potentially space. We don't work in that area, but just to sort of say that this is also some of the technologies are really driving towards how can you manufacture things, you know, when you really have a very, very limited importation and supply chain. And we're kind of somewhere in the middle where we're trying to go from this centralized setup that we have at the moment to something that's more distributed. And so we're really developing these DNA toolkits, which I'll talk about today. We'll also have a current project looking at developing open source strains. Most of the bacterial, yeast, and other strains that we use for manufacturing proteins have some encumbrances, even though they're actually, the patents expired a long time ago. So we've got a current focus on that. And then, as you'll see, we're always thinking about how we can go more frugal in some of the biomanufacturing, especially for individual labs that want to use these protocols. Of course, a manufacturer that is potentially scaling up, you know, there's kind of scope there to do more, but we really try and be, you know, equipment free as much as possible. So the core of what we've done, I guess, since sort of 2018 through 2022, and we still continue to expand this work, is looking at DNA toolkits. And so some of this was, you know, has been very embedded in the synthetic biology community for a very long time, that we develop DNA parts and components. And we just heard about how to build logic circuits in using DNA. And so this kind of engineering thinking of having, you know, components that you can put together in modular ways, it's very classic core synthetic biology fare. So the idea of having a DNA toolkit is not really a new one. But what we did was we screened all of the IP on these tools such that everything that's in the toolkits, we know the patent is expired or there never was one, and so it's available for use. And we started off developing the open enzyme collection, which has over 60. Right now we're at 81, and we're about to go through another round of synthesis enzymes. So useful enzymes for the lab that you might use on a daily basis: polymerases, ligases, reverse transcriptases, and then also a number of other you know, regulatory elements, tags with a special focus on kind of lower cost purification materials. And we distributed these with the Free Genes project at Stanford. And you know, just to mention, part of what we've had to do in order to kind of achieve this level of open distribution is also develop some new legal tools. So this collection is distributed under the Open Material Transfer Agreement. So we have 300 interchangeable elements, many. I think we're getting up to 400 now after the latest round. And so when we send these out, typically institutions require a material transfer agreement to be signed. Usually that says you can't pass this on to other people and you cannot use it commercially. Obviously that goes against what we want, because we would like these to be distributed as broadly as possible. And so the Open MTA was an effort from Linda Cale at the BioBricks Foundation, myself, many researchers on Open Plant, this UK synthetic biology research centre that was also trying to do synthetic biology tools, open source synthetic biology tools for plant biology. So we developed this legal tool that's basically very similar to the normal MTA, but it just says that you can pass this on to your neighbour, your neighbouring lab, your colleagues, whatever you like. So redistribution is totally allowed, and also that you can use it commercially. Industrial use is perfectly fine, and all we require is acknowledgement of the source of the materials in publications. And we published this in 2018 in Nature Biotechnology. And you know, now we've seen quite a number of Adgene have taken it up. So there's now not just our own plasmids, but other people are depositing under Open MTA, which is great. But the point is basically, if you receive our DNA, you can use it for what you wish to use it for. And we now have all of these collections, which we're building out. So we have now 2 open enzyme collections with 81 genes for these kind of workhorse enzymes, commonly used reporters, the open yeast collection, and a number of others. So we're distributing these. We know that they're in about 500 labs in around 50 countries, which is great. But are they being used is the big question. And how do we ensure that they get out of the freezer and actually onto people's benches and being useful? And so we've tried to build communities around projects and around kind of broader network initiatives. So the major one is Reclone. I'm going to skip through the sort of details because we'll come back to those later. But there we're starting regional reagent hubs at the moment in Argentina, Ghana, and the Philippines, where the DNA is situated and can be distributed more locally. We have a community, now a forum of around about 150 researchers, and we run monthly community calls and a number of practical hands-on workshops annually. And we've got a repository of protocols on protocols.io and a lot of kind of open resources for people to use those plasmids. Still a work in progress, but these are the people that are making it happen. And so just a little sample of kind of one of the technology kind of approaches that we've taken. So one challenge we had, particularly in working with colleagues in Africa, was that in labs, and this is particularly in East and West Africa, so I'll give an example of some work we were doing in Senegal, but many labs have not done recombinant protein expression before, which meant that they didn't have large shaking incubators and large centrifuges. And so we needed to find a way to produce proteins without those pieces of equipment. And we were tackling a different challenge, which was induction of the protein expression is usually with a chemical called IPTG, and it turns out it's not as stable as we thought it was, and we were constantly having to ship this stuff out. We already knew that it's a derivative of lactose, and we also knew that ubiquitously you can buy milk powder in any supermarket pretty much anywhere. And so we decided to see if we could use milk powder to induce expression. It turns out the answer is yes. But one of the problems is if you use liquid culture and you put milk powder in it, you can't tell if it's contaminated or not because it looks very cloudy with the milk. And so we were talking to a colleague who said, oh, well, you know, a person who works with bacillus, and said, well, why not just put it on plates? Because bacillus are quite happy on plates. Maybe E. coli will perfectly happily express protein just on agar, and then it doesn't matter if the media is cloudy. And so that's exactly what we did. And so hopefully this video will work. But we now have a plate-based expression where we put auto-induction agar media in a Petri dish. And then we just plate the cells the night before, pick them up the night after. There's differences depending on which media you use. So you ideally want a very rich one. And then effectively, you can just scrape the cells off and put them straight into a tube with no need for centrifugation. And we've now changed that protocol, so we don't use blades. We use, you'll see in a second, a glass slide, which is much easier. And it works. It's maybe not quite as much as liquid media, but we don't need as much. I mean, we have very low volume. So we have very low volume and kind of high yield enzymes. So one normal size Petri dish will give you thousands of PCR reactions. We've shared this protocol online. If anyone wants to go to protocols.io, you can try it out. And this is an example. Here we've added a reporter. This is a DNA polymerase that we're expressing, and we've added a pink fluorescent protein that's actually visible to the naked eye. And you can see you can just very easily scrape this stuff off the plate and go forward with your experiments. We can scale it up with big plates. And we're now working on training quite a number of high school and university lecturers in Latin America because it's just a very easy protocol to quickly make some enzymes. And this ties into another protocol where we don't purify the DNA polymerases. It's called cellular reagents and was developed in Andy Ellington's lab at the University of Texas, Austin. And then we further worked with Andy to try to even further bring down the cost of the protocol. But you don't need to purify, basically. You just collect your cells, wash them, dry them down, and add them straight to PCR reactions. And as long as you're not trying to amplify E. coli DNA, it works very well. Just to add that I also have an interest in low-cost hardware and open source hardware. And so this is also important. And just two examples of what we've been working on in that space. Franquero here designed this qubite system, which was published in PLOS Biology last year. And so this is effectively a kind of €60 materials cost tiny fluorometer for an 8-tube strip. And the reason we're interested in this is because we're now developing a whole suite of quality control assays. So once you express using our open plasmids, your enzyme in the lab, you only need this very cheap instrument to actually test that they're working. Obviously some reagents as well. I'm not going to pretend that any of this is, no cost, but it's certainly less expensive than having to have a qPCR machine on hand to do some of these assays or a plate reader. And so we're kind of now putting together protocols that then have an endpoint of testing the activity in something like this. And Fran is kind of working with a few manufacturers now to try and get this available for, obviously not 60 euros because they also have to assemble it and pay people, but certainly a more affordable price. And it has a kind of similar, you see here a comparison to a commercial thermocycler. And for the type of assays that we're running, it's very similar in terms of profile. Obviously, I can't guarantee that for a very sensitive assay, you're going to get the same results as for a several £1,000 instrument. But we have to think about the needs of the experiment at hand. Another example is the open flectroscope, which is a low-cost microscope, 3D printed with a Raspberry Pi camera. We've been taking some quite nice pictures of protoplasts with this microscope, and we're now building, based on some online designs and some working with a few providers of this particular instrument, to try and do a fluorescent version, which should hopefully bring the cost of having a fluorescent microscope through which you can view GFP expressing protoplasts down to less than about $300. That's the idea, and it's also packable in a suitcase for training purposes, which would be great because I will go on to now that we do a lot of training. And so we're really keen to have kits that we can readily distribute for training purposes, which is why having microscopes you can pack in a suitcase and take anywhere would be very helpful. And so we've done a lot of training in many places around the world, so we're quite used to landing in a lab, not really knowing what's there, and delivering molecular biology training. The point here is that we always try to build this into a train the trainer program, and a lot of post-training support with WhatsApp groups so that people who attend the training go away with the plasmids. So we teach people how to make DNA polymerases and other enzymes. They leave hopefully at least with the DNA and the protocols that they would need to go back to their labs. And as I said, we try and adapt the protocols so that they're accessible to as many labs as possible. It's never going to be universal, but we try our best. And then usually they're added onto our kind of tech support WhatsApp group. So if there's any problems, they can kind of send photos of their gels that aren't working or whatever, and we can troubleshoot together. We're still building this out and sort of learning as we go and developing the courses better, but we've done a lot now. So we have some experience of what works, what doesn't, and we can adapt it as we go. Okay, I haven't talked much about plants, so I'm very quickly, because I'm conscious that I don't want to run over time, what's the plant part of the work? So the main thing that we're working on is with the Grow More Foundation, which is a US nonprofit, who had previously, before we were in touch, developed something called the Enable Gene Editing Toolkit. And so this is a series of plasmids that allows for quite simple molecular biology processes to put your guide RNA of choice into a plant of choice for either transient or stable transformations in both monocots and dicots. And so in collaboration with Kate Creasy-Craner, who is the executive director at Grow More, and Florian Hahn, who kind of developed a lot of the designs for the plasmids, my postdoc Birhan Abati has been working to test these out. And we also collaborated with the University of Milan, who've been fantastic. So we put together a preprint. We're kind of planning to publish this. We're trying to develop a couple more case studies of using the toolkit, especially in orphan crops. So if anybody's interested, please get in touch. And especially also in the context of working in an African lab. Birhan is from the Bio and Emerging Technologies Institute in Ethiopia, and we'll be kind of working with them as well. But that's the idea. So we put the plasmids into AdGene, we've put the preprint out there, and then we're kind of just developing those last final pieces before we do the full publication, and always looking for people who might like to try it out. So do get in touch, as I mentioned. I won't go into the details. Many of you who do plant gene editing will be more familiar than I am with some of these pieces. But from an accessibility point of view, the goal is really enabling more labs to use plant gene editing by simplifying the initial molecular biology as much as possible. It is not the only toolkit that has this goal. There are several of them, but we've had in mind very specifically low resource labs, and the kind of addition to that is thinking about how can we distribute this without the cold chain issue. So our work in progress at the moment is developing ambient temperature shippable toolkits that contain the plasmids, which we know we can ship ambiently anyway, but also we're currently testing lyophilized enzymes that we can package so that people can get one plate, which enables them to do a bunch of reactions, but then also the plasmids to express more of those enzymes if they have the kind of interest and capability in their own labs. I've kind of grayed out the Cas9 plasmid because there are some IP issues with that. So that may not be distributed with the toolkit. There may have to be a separate order made by the receiving lab to get that themselves. But that's the idea. So we want to support plant gene editing training and research in labs that experience supply chain issues by kind of packaging everything together, but also giving people the tools that if they want to make their own package, they're very welcome to. This is, again, an open initiative. We've already piloted the hands-on training with the toolkit in Ethiopia. We learned a lot through that, and we're still developing some improvements based on it. But we got a lot of really good feedback from some of the students who had not had a chance to really work with plant gene editing, and also some of the molecular biology techniques like producing competent cells, and really getting the hands-on experience for the theory that they'd learned. We were part of a bigger initiative around gene editing of sorghum. Unfortunately, the funding for this was discontinued last year in the big wave of funding that was discontinued across the world for international aid. But just to say that, this is the kind of the vision piece, is that we can develop these hubs that can support this type of work in partner countries, and that it really kind of links to economically important work with crops and plants. And this is just the team from the Smarga Strike Sorghum for Africa project. And that's it. This is the way that we work, technology, community, and enterprise. And yeah, thanks to everybody who's part of the group and has contributed to this. And we welcome any questions and collaborations. So keep them coming in the Q&A of the webinar. Thank you very much.
Great, thanks, Jenny. That was amazing. Very different type of talk as well to usual. So thanks a lot. Please put your questions in the Q&A. We'll go quickly over to our last speaker, Rachel. So Rachel is a professor of philosophy at Wageningen University. So she brings a very different sort of interdisciplinary perspective to history, philosophy, and the ethics of life sciences. And she's particularly looking at how science knowledge is produced, how values influence scientific policy and practice, and how public engagement can guide sort of decision-making, ethical decision-making. So Rachel's talk today, as you can see, is going to be around social science insights for researchers in crop gene technologies, the importance of concepts and framing. Thank you, Rachel. Oh, you're on mute, I think.
Okay, sorry. I thought Isabel might do that. Thanks so much, Catherine. I want to talk about framing, and although in the overarching title there was this idea of acceptance, that's actually something we find quite problematic. And so I want to try and avoid acceptance talk, but we'll get there in a minute. I would just want to start by acknowledging the Garna people. This is something we do traditionally in Australia. I'm not sitting in Australia, but this research was done in Australia. And so we always believe that it's important to acknowledge Australia's First Nations people, their relationship to country, and our respect for their past, present, and ongoing connections to their land and their cultural beliefs. You'll see in a minute, as part of the broader project that we're doing, there is a component on Indigenous farming and its connections, actually, and I need to put them in touch with the previous speaker. So just a bit about me. As noted, I'm a professor of philosophy. I do use a lot of social science techniques in what I do. What we try to do in a range of the projects that I'm involved in, both at Adelaide, where I was previously, and currently now at Wageningen, which, as you probably know, is a big life science university, but also in collaborative projects around things like open science and agriculture. We try to use a range of analytic but also empirical methods to look at ethical, social, and regulatory issues in food and agriculture, and in turn ask broader questions about norms in science, understandings in science, and how to engage and involve publics in science. And you'll notice I'll use this word publics in the plural because this is something from science and technology studies that signals there's no one, unwashed general public. People belong to multiple interest groups, multiple kinds of parties, as we all do. People are scientists, but they're also parents, they're citizens, they're perhaps environmentalists, other things. And so when we look through all of our collaborative projects at these issues, we always try to look beyond kind of a narrow conception of who these publics might be. The other thing I want to call out in this slide is that this work is part of what we call the Food Values Research Group. And again, the plurality, this recognition that people come to food, agriculture, and the science behind it with a pluralistic range of values. And we try to be open, particularly through the empirical work, to hearing about those values. And I'm going to talk a bit about the training center in a minute. So why social science approaches? Underlying all of this is a deep belief that science and technology always occur in a context, and that it's super important to understand that context on its own terms. A second, maybe starting assumption, but we've also found it in our research, is if you're interested in social acceptance, and I do understand scientists and tech developers are interested certainly in not ending up with a public that's opposed, this isn't going to be generated simply by telling them how it is or teaching them about it. Generally, the public's willing to sort of say, y'all are experts. Sorry, I still do have my American tics. That scientists in general are experts about certain technical things. They're not interested in the details, for example, about the difference between gene editing and genetic modification. But they are interested in a whole range of issues that'll come out in what I present. And therefore, we need to be careful not to continue what's been known as the deficit model, which sort of says people are empty vessels, fill them with knowledge, and then they'll come along and believe in the science or endorse the technology. That isn't an accurate picture psychologically, empirically, or really ethically. And finally, we found in our research that publics don't tend to be simply for and against. There are some loud publics that are against certain things, but they tend to be fairly small and getting smaller. The details matter. And I think with climate change and other things, we're really seeing that shift. So our methods, as you see in the box, include empirical data collection, largely qualitative. We do some quantitative, but we're interested in the reasons why, not just what people think. We then use a range of different kinds of analysis, thematic, discourse, rhetorical, to look at what's going on in that data. We also, in some of the projects within our group, look at the legal and regulatory aspects. And I should declare I've been on the Office of the Gene Technology Regulators in Australia, Ethics and Community Consultative Committee for in different forms for about 15 years. So I work closely giving advice to regulators, even though I don't myself come from a legal background. And in this domain, then we use ethical analysis, but also public engagement to think about the role of regulation. And I'll talk about that in a minute. So I want to give you some key findings from our earlier projects over the last, say, 10 years. I've already said people's concerns, particularly about GM, but even GE and biotechnologies in general, are not usually about the specific technology or about the science. They're interested in these other questions about health, control of the food system. What are the risks? What are the benefits? Who are the benefits for? And more generally, how they understand good food and what it means to have a good life. So that's very philosophical. We're trying to look at these social cultural contexts within which these issues are made meaningful, any issue about biotechnology. So we've done a range of things. This includes gene editing in livestock, new breeding technologies more generally, and people's understanding, say, of labeling and disclosure. We've done empirical work looking at subgroups like women, scientists, and so on. I've already said deficit model is not a good idea, but the positive side is needing to foster transparency and trust. And I think a lot of the things talked about in the last talk are really important ones for transparency. And finally, this is critical for scientists and industry. It's as critical as getting the science right. You need to get this domain right. So the new project, relatively, we're nearly at the end of it actually, but the newer project out of which the papers in this special issue come is an Australian government-funded ARC training center for future crops development. We're really fortunate that in this center, responsible innovation, as it's called, which sort of is a label to capture everything I've been talking about, is one of the programs alongside developing applications, doing the sort of lab-based work, and looking at applications in the field. So this is a joint venture between Adelaide, what's now Adelaide University, and the Australian National University. And our platform, if you will, or our program is this responsible innovation. One of the key things, because we're a training center, is that we have developed ways of approaching responsible research and inclusive innovation, teaching and engaging around the regulatory landscape and all its interesting changes. And then also separately from those things, science communication, which includes engaging, but how do you talk about the science that you do in a way that's ethical, but also clear, and so on. We also do research in all of these spaces. So that's the nice thing about a training center. These two things occur side by side. As part of the research side, then, we're very lucky to have a series of really fabulous PhD students, a number of whom are on this webinar, looking at different aspects of the questions associated with so-called future crops. I'm going to talk about two of the projects in more detail because they're the papers that were published. But just to note, the one in the middle, the gray, Lucy Dara at ANU, is really looking at this question of framing. How are questions and discourse talked about, both by scientists more generally, when they promote or publish about agricultural biotechnology. Down on the left in the gray, Ava Wilkinson at, sorry, that logo's the wrong one, but what was University of Adelaide, now Adelaide University, is looking at what's called market shaping. So how, instead of seeing the market as passive, scientists, industry, and other stakeholders can contribute to shape this market for what will be sort of the next wave of crop technologies. In the middle, Jamie Puro back at ANU is looking at knowledge production and understandings about responsible innovation in particular. And finally, as mentioned in passing before, Audrey Henry at Adelaide University is looking at indigenous knowledges and multi-species justice in crop science research. So we've got a wonderful sort of mix of projects. We're going to focus on two, which were the papers that were published in the special issue. So the first one here is Mikhail Ivanov, who's a legal scholar, but also deeply interested in the intersection between regulation and societal expectations. And for this paper, together with Emily Buttle, who is the postdoc on the project, we looked at the rhetoric that we hear, both in passing. I hear it all the time when I talk to scientists. We hear it a lot in our regulatory engagement meetings, is the idea that regulation is a bottleneck. It's getting in the way of innovation and progress. And to explore this then, we looked at published and grade literature to try to push a revision of our understanding of the role of regulation. So in some sense, trying to encourage people to abandon or at least go beyond this bottleneck vision of the relationship between regulation and plant gene technology. So bottlenecks, sorry, that's a terrible fuzzy illustration, but you'll get the idea. Bottlenecks in general, you can think about your favorite metaphor. We talk about them in detail, but basically they're a point of congestion in a production system that stops it from functioning at full speed. You can think we use the example of traffic where they look for bottlenecks in traffic, then they look for mechanisms for cleaning it up. In our case, where we're hearing people talk about bottlenecks is this. We have crop R&D, which includes all the way back, even to the first talk, lab-based research, and scientists seeing that regulation is getting in the way of getting the science and technology out into the market, be it through testing, but also sort of commercial use. And therefore, regulation is an impediment. Regulation gets in the way. It's a point of congestion that ideally we would just, quote, get rid of, right? And sometimes you hear people say, oh, we need to get rid of the bottleneck. Sometimes people go all the way to saying regulation's the problem. We need to get rid of it. And yes, this is sort of casual talk, but it's something that runs very deep. So what we want to do in this paper was this reconceptualization. Think about R&D as, regulation is a critical part of the R&D process, that it actually allows diverse publics to think about research and think about whether a biotech is desirable and beneficial beyond the lab or whether it's likely to be. And regulators are doing that in many ways on behalf of the publics, enacting their mandate, which is different in different places. What we think is regulation is a positive because it allows for this reflection on innovations that in turn can make its use more responsible. What do we mean by more responsible? More socially acceptable is part of it, but also more transparent, more legitimate. In democracies, we think about justification and legitimacy. And the regulatory process allows these innovations to become part of the dialogue in these spaces. And finally, then, why do we have regulation at all? Well, regulation is part of a broader social system. Regulation is there to shape, and this is a very philosophical / legal way to put it, agents' behaviors. And here we mean scientists, people in industry who might be planning the market for the future, farmers, and so on. Anything regulatory isn't there as an impediment, but it is there as part of a system that's designed to shape people's behaviors to make sure they're aligned with social expectations. So think about your favorite example, traffic, things are regulatory, we put things in place. They're not necessarily meant as policing as such, but they are meant to shape behaviors to try to make sure we're constantly living jointly in a society where we're fulfilling others' expectations. So therefore, you can't be a rogue agent or make decisions on your own because this is a social space. There may be things you can make decisions on your own, but regulation is in the domain where we really do want this shared space of acceptability. Now, you notice here, some people are going to be unhappy, right? I don't particularly like the speed limits in certain places, right? But nonetheless, I might abide by them because I realize this is part of this broader set of social expectations that foster safety, other things, right? And so too, we will have loud voices, and we do. Perhaps activists who think nothing in this space should be done, and perhaps scientists who think everything should be done and they should be allowed to do what they do. And here I think I really want to call out debates over academic freedom miss the point. Okay, it misses the point about regulation and what it's about. First of all, academic freedom, a good thing, but it's not a be all and end all, and it's not an unlimited thing. And conversely, most scientists are funded by publics, right? The public, government in most cases. And therefore, there is this sort of implicit, I don't want to use the word contract, but, and we avoid the term social license, even though it's super popular in Australia. There's an implicit agreement that they are going to act in some way aligned with the social good. And regulation is part of this process. Okay, so in this paper, after making that whole series of arguments, motivating it using more examples, and I encourage you to read it, get in touch with Mikel, but we raise the following questions to consider. We don't tell scientists or industrial actors what to do. We do think there's a question in some way for us as people looking at regulation, but also regulators. We recognize that a lot of regulation is not as innovative or agile as it might be. It hasn't kept up with change. This is not to say that, for example, gene editing should just be assumed to be X, Y, and Z, that the scientific answers are always correct, right? But we recognize in some cases it's inefficient. And that is due to a range of reasons, including lack of funding for our regulators. But there may be ways to be more innovative and more responsive, more flexible. And that's something Mikel's thinking about in general. Second, we want to think about how ag genomic scientists, most of you I expect on the call, can be more engaged in regulation, not just, at the point at which they're fighting against it. And more generally, how can this engagement be more productive given these longstanding conflicts, for example, with activists? A broader question is then how to involve a range of people early in technology development and how that might have effects on regulation. This is part of what we're trying to do in the center through our research and through our training, is bring together the social science and the tech development and the research. But how could this be embedded more broadly? And then finally, how to train scientists to understand all of this, including how they can engage with regulation. So a second paper and a second project, Chris Wenzel, pictured here, in the special issue, looks at the question, which seems like a simple one and gets harder very quickly, which is what is sustainability? Again, we noticed that sustainability was being used as a rationale a lot for research. And that's fine. There's nothing wrong with that in itself, right? And we fully understand, we write grants too, that you need to make reference to these broader things, including sustainable development goals. But what we wanted to do was see how people defined it, particularly because sustainability often is talked about in 3 different ways: economic, environmental, and sort of rural or social sustainability, right? The paper gets into this in more detail. So we used a structured review of literature on GE crops in a defined period to see how people were using the term. We're also super interested to see how they measured whatever their research was against a metric of improvement to sustainability, and expected that we would get papers that sort of said, oh, this is the mutation that we're developing. And look, if this were to be put into place, it would have this and those effects on crops and therefore contribute in this concrete way to environmental sustainability. Something like that is what we were looking for. Why do we care? Well, because sustainability is one of those terms that can be used in a vague way, call it sort of greenwashing. It's super important to make clear what you mean, not in the least part because there's multiple definitions. And, providing at least a hint of the cause and effect relationship between the research and a claim about improvements in sustainability is critical, because otherwise it's just sort of a buzzword we're using in our grants. We went in with an open mind, okay? But what we found was when we coded people's claims about this, there was almost no definition of sustainability or engagement. It was just assumed everybody knew what was meant. And there certainly were no metrics, right? And so on this chart, most of the articles we found, fared pretty badly. There were a few that had attempts to either define or engage or tell us how the research measured up. So some conclusions, really important. Our intention was not to criticize specific articles. And we did have a few people sort of say, well, where did we fall? That was not the point. We're trying to raise awareness about these issues. Again, we were interested in standards of evidence. We weren't looking just to critique. We detected is you might have different hypotheses. Oh, we used to kind of write in this vague way, we've gotten better. That was not true. Only bad journals publish these things. Not true. Some disciplines are vague. Not true. Those people over there, think of the last talk, are not using good. That wasn't true. What was really interesting is the claims tended to occur in the abstracts and the introductions and the conclusions, and not in the body of the paper. A hint here that we, could have done a different study that would have been really difficult, ask people for their grants and see if they simply pick up their grant rationale and slam it into their introductions. My inclination is to think this kind of wording is being picked up from other places. So just a few quick lessons and I'll tie up, Katherine. I think it's really critical to think about your rhetoric, and particularly when publications travel into public arenas. You engage with the media, but anybody can access a lot of this stuff. And, when you make claims, somebody's going to wonder whether they're good claims. And so we have the micro claims in the articles, which I assume the peer reviewers have looked at, about the specific mutations, specific intervention, whatever it is. But the macro claims, the framing matters. And I think it's important to reflect on how you do it. I think there needs to be more understanding of best practices for sustainability claims. And this may go to scientific publications and reviewers needing to say, really? How does this tie? And we could have written a similar paper, frankly, about food security. And I've got data that looks almost exactly the same. So this is a call to make claims more robust and transparent, but also think about collaborating with environmental researchers, with people who can help with those metrics. So these are not quick conclusions, but trying to tell people that biotech is good, safe, or harmless is not going to be useful. You need to think about the purposes, who might benefit, and make all that really transparent. Regulation is your friend. You're never going to get uniform bans, probably. You're never going to get completely open markets. Regulation can create dialogue. We need better standards. We need better arguments. And we need more recognition that people do have equal standing when we're talking about policy, the common good, and what we do for society. Science doesn't know best about all of that. It makes really big contributions, but there's lots of questions that are these higher level decisions. And the main outcome needs to be some sort of legitimacy, even though you're still going to have actors who disagree. And so thanks for your attention. These are our team. I'll close.
Thanks very much, Rachel. That was also really, different and interesting approach. That's good for us all to think about. So thank you. Okay, so in the last few minutes, I can see that Adil's been very busy answering questions on the chat, which is the advantage of being the first speaker. I think Jenny's asked one. If there's any more questions, please put them in the Q&A. There's one question there, but it's very broad about tools with AI. I think that's probably better something tackled offline with specific people. But any other questions, please put them in the question and answer. I had a couple of questions, if I can, seeing as there's nothing there at the moment, I can do. So, Adil, I was interested, you had a whole range of promoters of different strengths. How did you make those? Was that random mutations, or had you designed those?
That's a very good question. So what we had done, so we had taken like non-guide RNA binding sequences for the Cas9, which had been validated in different systems, for example, yeast and human genome. So we knew those have been validated in the lab and from some other published work, hoping for they wouldn't be like in the plant genome. And then we simply replace the native sequences upstream and downstream of the tartar box to engineer those promoters, because plant promoters are AT rich and the PAM sequence for the Cas9 is NGG, so it's quite hard to find those ones. So that's why now we are moving from the Cas9 to other CRISPR proteins to expand the toolbox.
Great, thank you. And I'm going to keep going, seeing there's nothing there. So Jenny, I mean, I guess you alluded to this a bit. I mean, your work is, it just seems so important, but presumably, I mean, is it hard to get funding? I mean, how open are national sort of, they're so focused on, industrial sort of commercial things or our fundamental science. I mean, is it really hard to find funding to support this sort of type of project?
I think it depends. I mean, obviously, there's been a massive shake-up of generally international development focused funding in the last 12 months. So that's been challenging. And that's had ripple effects with a lot of philanthropic funding, which is where quite a lot of our support came from. But having said that, the work that I mentioned on open source strains, so this is E. coli and yeast strains, is actually supported by the UK government, because the focus of that project was open source tools for UK startups and SMEs where they need these resources and they don't necessarily have tens of thousands of dollars to pay for like commercial evaluation licenses, for example. So I think there are aspects where, and we're not anti protection of IP, right? But there are some cases where, these tools are decades old and it's not actually the formal patents that's now inhibiting their use. And so I think there's a few areas where there's a real interest for higher income countries and kind of, commercial entities in general. And I've spoken to a lot of biofoundries and kind of contract companies. They're also very interested in this because they want foundational strains and cell lines where they can then develop innovations on top of that. So I'm optimistic, but it does require a reframing. And then, of course, I think to Rachel's point as well, we always have to be careful about like how we're framing the work that we're doing in different contexts and the benefits and impacts. So that's a constant sort of dialogue and challenge, I think, in our work as well.
Okay, no, that's really, that's really clever to sort of, as you say, you can reframe it in terms of, as you say, SMEs and access, engineering, biology and everything. Access to SMEs is a huge government, focus. That's great. And Rachel, so because I was interested in your responsible research and innovation, because I mean, certainly in the UK, we have to address this in every grant proposal now. So, and I guess, are there good, I don't know whether this is appropriate or not, but are there good sort of, I don't know, focus toolkits or things that make you, like you've got your questions that come out of your papers. Is there something, I guess we need to look at your papers, or have you developed resources that sort of help scientists answer? You're on mute. Sorry, just one sec. There you go.
I think Isabel and I are dueling on muting. That's part of what we're trying to do, through these papers, but more generally in our center, is to develop a range of kinds of toolkits that can help. And here, when I say trainees, I mean, these are scientists themselves. I mean, these are things that also need to happen from the top down, because of course, lab leads set the standards in labs, journals set the standards and so on. So I'd want it to be both sort of the training of new people, but also it needs to come top down. And similarly, the kinds of principles that Jenny's interested in, right? One of the things I'll put a call out for is that we will have an open access book, probably by the end of the year, that provides accessible approaches to responsibility in agriculture biosciences more broadly. And this is designed both for students to use in the classroom, but also for scientists themselves to be able to use. And that's something that Emily and I and Mikael actually are doing together with a colleague at Nottingham, Kate Miller. And I think that'll be a really good book because it's also designed kind of not so covertly or not so sneakily to teach scientists, right, as they're teaching. So the vision is, you have one ethics unit, maybe one lecture. You could use a reading from this book to stimulate discussion, and then there'll be but they're real research articles, so it also has that kind of deeper edge. Yeah. So watch the space, get in touch with us. Yeah.
Great. Well, thank you very much. I think, yeah, I just want to end up in the last couple of minutes just to say a really big thank you to our three speakers. I really enjoyed it, and you can see from the chat that there's been a really great reception to your talks, and we've still got nearly 200 people signed on at the end. So I think it's a great reflection of the work you're doing. So thank you again, and we look forward to seeing people again at the next webinar in May. Thank you very much.
Thank you. It was great.
Thank you.
So thank you, everyone, and let's see you soon. We have a new, we will have the Global Plan Council will have a new webinar next month. I will send you all the information. And I just wanted to say to the speakers that normally I'm very satisfied when a webinar ends, but this time especially, I really enjoy the different talks, and I also enjoy very much the interdisciplinary perspectives coming from each of you that are from talking about the same, but from the different perspective, and I think it was very enriching. And because many of you, I think the three of you are designing tools, handbooks, or useful resources for the plant science community, I will be contacting you because in the Global Plant Council, we have a specific section for that. So to make it more available and all everything put together for the plant science community at a global level to be able to access and use it.
Fabulous.
So thank you so much.
Great. Thank you.
Thanks. Bye-bye.
Bye.
Bye-bye,
Thank you.
everyone.
Bye-bye.
Thanks, everyone. Bye.